at the HND level. You already know me. Maybe a few of you don't know me. You are now, maybe you finished school a long time and you are now coming. I don't know. Or maybe all of you, you would have met me already at the HND level. So my name is Lawrence Nubienyo, and I'll be taking you in SBT 401, General Biochemistry. What you have on your screen is the course outline for the semester. The semester, I think, is about 16 weeks or 15, 16, thereabouts. Maybe when you take out the week for, they have maybe a week for revision, a week for mid-semester, something like that. So, maybe you have like 14, 15 weeks. But generally, my courses, what I do is that I try as much as possible to plan within a period of 12 weeks, taking into consideration sometimes some holiday will come in and anything can happen, so that whatever it is over the semester, we'll be able to finish with our plan, or everything that we plan to do, we'll finish it. So on the course outline, we have 5 main objectives under the course. The first is on fundamental enzymology, so some studies on enzymes. And then we go forward to look at metabolism of carbohydrates. Then we look at metabolism of proteins. And we look at metabolism of lipids, that's fats and oils. And then metabolism of nucleic acids. So generally, this is what we are going to do. Under each of these objectives, I try to break it down into 3 sub-objectives. Call it some performance criteria, where we'll be discussing the structure of enzymes. We'll look at classification and nomenclature of enzymes. We'll continue to look at coenzymes and cofactors. And then we look at some mechanism of action of enzymes, as well as inhibition of enzymes. So then we would have finished with that part in fundamental enzymology. Then we dive into the intermediary metabolism of the biomolecules. So we first take, say, carbohydrates and look at the processes of glycolysis, the TCA cycle or the Krebs cycle. And then we look at processes of glycogenesis, glycogenolysis, and the pentose phosphate pathway, or the hexose monophosphate shunt. Some of these you would have seen already. Those of you who did HND, I'm sure you had done some glycolysis, the Krebs cycle, which is metabolism of carbohydrates. Now, I don't know if you did glycogenesis and glycogenolysis. Perhaps you did. But it means that for you, it's going to be some form of revision. Or maybe we'll pay attention to some very things which when you were doing your HND, you may not have paid attention to. So obviously there will be some new things that you can learn from there. We'll continue to look at metabolism of proteins. So the breakdown of proteins. And then we look at processes of transamination, deamination, amino acid oxidation. We'll take some specific metabolism of amino acids such as glycine, phenylalanine, and tyrosine. We'll talk about them briefly. Then we go on to lipid metabolism. Here we will talk about biosynthesis of lipids as well as oxidation pathways, the alpha, omega, and beta oxidations of lipids. We'll look at those. And then lastly, we take the metabolism of nucleic acids. This actually, we can call it metabolism of nucleotides because these are the basic units of nucleic acids. So we'll look at metabolism of purines and the pyrimidines, looking at what we call the salvage pathways and then the breakdown catabolism of the nucleotides. And we'll speak briefly about some health implications that are related with metabolism of nucleic acids. So that's our plan for the semester. The last part, you are very familiar with it, which is your assessment, our activities over the semester, attendance, some assignments that you do, or anything that we do over the semester that we can assess, add up to give us 40 marks of continuous assessment. And then the exam will be over 60, so that when you put all of that together, it will give us the 100% for the course. So that's our plan for the semester. And I don't know if there's anybody in the class who is mature student, or maybe he's finished school for a long time, went to work for some years and came back. It's possible. Okay, more people have joined. I think you are now 17 people here. Perhaps more people will join over the semester. I want to say that for biochemistry, I've seen the frustration with students all the time talking about biochemistry being difficult and all of that. But as a matter of fact, all you need to do is to have some patience for yourself. I mean, it's an advice I give to all my students. If you are trying to do something for the first time and it's difficult, it doesn't mean it's so hard that you can't do it. The person who is teaching you is a human being. So if he understands and is teaching you, it means you as a human being, you can also understand it. So it just requires you to sit down and do your part of the work. I will do my best to give you what you need to go through the course. So I liken the work of a lecturer to an analogy of, say, for instance, we are making a journey. I happen to go ahead of you and I'm already at a destination. So I'm only telling you that when you are coming, when you reach this point, be careful. There is a bend here where you need to slow down. There's a place here, there's a valley, there's a hill, there's a pothole. Just telling you, how to navigate your way through. But in the end, you have to do the navigation yourself. So I would say that all you have to do is to sit down. When I give you all the guidelines, everything that you need to study, sit down, and then you'll be all right with it. So those are a few words that I'll tell you before we continue. Now, once again, for those of you who have now joined, if you have anything to say, any comments, any contributions, any questions, you can write in the chat. Alternatively, you can jot these things down. After the lecture, usually we'll have like at least some 30 minutes. I will open the floor and you can raise your hand, and then I will enable your mic. When I enable your microphone, then you can unmute yourself and you speak, and I'll respond to it. So that's for those of you who are just joining. Okay. For this semester, I've decided to, give some bonus introduction to biochemistry in all my lectures because of experience of the frustration that I witness in students concerning biochemistry. So your class will not be left out. Very brief introduction to biochemistry in general. Even though some of you have just finished HND, I'm sure a lot of you are still relating to biochemistry as some kind of difficult thing. You have still not come out of that relation to biochemistry. So we'll have a brief introduction to biochemistry before we go into the main business of the day. Now, before I also want to say that I hear a lot of students also complaining about online lectures. That is difficult. It is this. So many complaints. Everybody has something to say about it. Now, I'm saying this because this semester, I'm not there in Ghana, so we are not going to meet face to face. All our lectures are going to be online. Attendance is going to be taken automatically. So those of you who are not writing your real names, people enter into the class with some funny names. At least in there for now, this class, I'm just checking. I'm seeing that at least you have some real names there. Some of you not full name. So the next time, I hope you can, put your full names, and it will be better to put your student ID there as well. I think so far there is only one person, Letitia Mensah, that has put her name and her student ID. The rest of you all is either just one name or just your name there. So if you can edit those things the next time, because it will be recorded. The attendance is going to be automated. So if you have those names and it doesn't recognize them, like if I cannot tell who is this person, is not in the list of students, then it will be going against you. So that's one thing. Now let's continue. I was saying that a lot of students complain about online lectures. Everybody has something to say. It is this, it is that, all sort of things. But I would like to say that online lectures actually gives you a certain opportunity that the in-person lectures will not give you. So for instance, now this lecture is being recorded. So at the end of the lecture, you have access to this lecture to watch it as many times as you can. That opportunity you will not have with an in-person lecture. So it's actually something which you should be embracing. Say, for instance, your class is not that big, but in some classes, large classes, that you have a screen in front and you have a number of people in the class. And yes, people who are in front, in the middle, can see very well and they can hear, but those who will be behind will not be able to see so clearly sometimes and may not be able to hear so well sometimes. But here is the case that each of you has an opportunity to be behind a screen, a device that at your own comfort, you can see clearly everything and you can hear. And even if along the line certain things don't sound well, you don't hear certain things, maybe because you have some network issues and all of that, the video is still going to be available, and so you can always refer to it. So it's an opportunity that I will encourage you that you should embrace it. The world is moving forward, so you don't get stuck to one thing. In so many universities now, some courses have gone completely virtual, like theory courses that students will go and do it at their own pace, and there will be a particular time that they take assessments and they go do practicals in addition to the theories that they do online, and that's it. It's not only like outside of Ghana, even in Ghana. I know, for instance, in Gimpa like this, there are some courses that are completely online. The students will have to do and the assessments, they write a report, and that's going to be used for their continuous assessment. So it's a thing that you have to get abreast with, and then don't be resistant to change. I tell you a story. This particular course, about 2-3 years ago, we are in 2025 now, so yes, I would say that's about 4 years or even 5 years, because that's like 2020. That's right after the COVID-19. At the time, ATU had not procured online teaching tools like Zoom or these Microsoft Teams for lecturers, but there were still restrictions of the number of students who could be on campus at the time. So that was actually the beginning of the introduction of alternating online with in-person lectures in ATU, because there had to be a control of the number of students that are on campus at the time, because the facilities will not allow for you know controlling the numbers in a class at a time. So the only way was that some people have to be online while some people are in person. And at the time, there was one student in my class who couldn't attend most of my lectures. But that student was one of the best of the class. I mean, because the student was he was sickle cell, and many times he will be in crisis and be hospitalized. But at the time, it's not like now. Now it's easy. As I speak, I know that there are people who are listening to me. It's natural. But that time, I had to get the software and record the lecture. it's like speaking to myself. And after recording it, then I send it to the students to watch. And the student came to me one day to say, he said, thank you for your video lectures. And he said, look at my notes. Even though she couldn't come to class, she wrote the notes as if she was in a class. Ditto, everything. And so it's about you sitting down, doing your part of the work. It's not about the lecturer coming to be in front of you every time talking. I'll talk. It's going to be I'll be with you for maybe uh, 1 hour, 30, 40, 50 minutes to 2 hours. But after that, if you don't go to sit down to study, there's no way that is going to stick. Like I say always, biochemistry, if you do it, have a jotter, try your hands. You don't just open the book and be flipping through it as if it's some kind of a picture magazine that you are looking at and think that you understand it. So I would say again, do your part and everything will be fine. Okay, so um, that that said, uh, I promise that I will give you some introduction to biochemistry from the beginning before uh, we go into the main business of the day. So when you look into the course outline, the beginning there was something on the fundamentals of uh, enzymology. So we'll do some fundamentals of enzymology. Before then, we will do some brief introduction, generally to see what biochemistry actually is, so that that fear that you always have around this course, I hope that it will be gone. So you begin to understand that actually it's not such a complex thing as you were thinking, but it's something that is systematic. You just have to take time and have some patience for yourself, and you'll be all right with it. Okay, so when you take the word biochemistry, clearly it's coming from two words, biology and chemistry. Now, even in the uh, primary school level, even GSS, SS, you studied biology is what, study of living things. At first, you did you did some distinction between things as living things and non-living things. And so biology was that subject that studied the living things. How about chemistry? Chemistry, you study chemical substances and then the interactions that they have between them. So what it means is if you say biochemistry, it means that chemistry in living things, which means interaction of chemical substances in living things. So you have biochemistry. In other words, some people even call it biological chemistry. That's what it means. Okay. Now we'll go step by step, break it down. What I've done is I've broken this down into like 5 stages to get to the point where biology and chemistry merge to give you what we are calling biochemistry. So stage 1, biology. You are talking about living things, which you generally you call them living organisms. Now, in stage 1 biology, it means that you take all living organisms and what do you do at that stage of studying living organisms? You categorize them. Is the living thing an animal? Is it a plant? Is it a fungi? So there you do what you refer to as classification of these organisms. That's when you talked about living organisms classified into kingdoms. You say kingdom Animalia, kingdom Plantae, kingdom Protozoa, all of that. And under each of them, you had phyla. And the phyla too will be broken down into classes. The classes broken down into orders, orders into families, families broken down into genus, and then in the genus you have the species. So you do this classification of the living organisms at that stage. How about chemistry? So chemistry, you are studying chemical substances and the interactions between them. And these chemical substances, what are they? They are elements. The elements that make up objects. And so here too, you do classification of the elements. That's what we refer to as the periodic table. So you are classifying these elements into metals, non-metals, but then the metals, you have some alkali metals, you have transition metals, you have halogens, you have inert gases. Even in that periodic table, you have some classification into groups and periods. You have some group of elements, you refer to them as actinides, some lanthanides, all of that. You do that classification with a periodic table. You move down to stage 2. Now for biology, remember in stage 1, you were looking at all the organisms and classifying them. Now in stage 2, you take a single organism. And what do you study at this stage? You are studying the organism to see the systems that make up the organism. You refer to that as organ systems. So for instance, let's say if you take the human being, you are looking at, say, digestive system. You are looking at what? Respiratory system, circulatory system, etc. And each of these systems are made up of organs. So if you take digestive system, for instance, you're talking about the buccal cavity, the mouth, joint to the esophagus, going to the stomach, duodenum, uh, small intestines, large intestines, all of that. You study all of that. You take the circulatory system, you study what's the heart, the veins, the arteries that are connected to them. Even the heart itself, you want to talk about, its structure, but even that, it will go to the next. So then the organs are made up of tissues, and the tissues are made up of some specific cells. Then the cells, you say they are made up of organelles. So you have a cell membrane, and then you have the organelles in the cytoplasm. So you look at that single organism at that different levels of the organism, the structure of it. In chemistry, when you take an element, what are the elements made up of? They are made up of atoms. And so you study the structure of the atoms of these elements. And when you're looking at the atomic structure, you say that you are looking at the nucleus. So every element has some specific number of protons in its nucleus and some neutrons. That's what is going to come together to give you the molecular weight of that element. And then you have the nucleus, being surrounded by shells with electrons. That's how we studied the atomic structure at that stage. Now, when you move to stage 3, so here you've gone into advanced biology, advanced chemistry. This time around, you are referring to the biology as anatomy and physiology. And what is anatomy and physiology? Anatomy talks about the structure of an organism, or say the structure of the organs and the tissues that make up the organism. And the physiology is talking about the functions of these organs and tissues. So you take a specific organ, like say the heart, then you begin to look at the structure of the heart. That's when you draw the heart, you start looking at the various chambers, the atria, the ventricles, the veins and the arteries that are connected to it, the valves, and the tissues that make up this organ. When you finish looking at that anatomy, that is the structure, then you now move and start looking at the function, how blood, moves in which direction, to what direction, through which valve does it move into this chamber and move out and circulate around the body. That's function. So each and every organ, each and every tissue, whether it is muscle tissue, skeletal tissue. So, anatomy is studied in such a way that like you are here, as you are there now, what you can see, like if you are doing anatomy and physiology of the body, you can see your skin. Under the skin, so you first you study the skin. I think you did some studies on that sometime. You draw the skin and you see all the layers and the components that are in it. And then, or even in some stages, you even look at not just the skin, but the skin and appendages like the nails, the hair. Then below it, you are looking at the skeletal muscle. That is also studied, the structure, different functions, which muscle is connected to which joint, moving this or that. After that, moving to the skeletal tissue there and looking at the different bones and things like that. So that is at that level of biology. Chemistry now is talking about interaction of the elements. Remember, you were talking about the atomic structure. Now you start to talk about interaction between the atoms. So it can be atoms of the same element, which means there will now be formation of bonds. Is it covalent bond? Is it ionic bond? Van der Waals forces, hydrogen bonds? You start talking about these things. Formation of molecules. Once there are formation of bonds, there can be formation of molecules. And there can be formation of compounds, that is bonds between atoms of different elements. And there can be formation of complexes. Now, you move to stage 4. In biology, you now move to the cellular level. So you start to study the structure of the cell, where you are looking at the cell is made up of the cell wall or the cell membrane, the cytoplasm made up of the cytosol and the organelles. And in chemistry, you're now talking about interaction between the molecules. That's when you start talking about chemical reactions. You talk about which type of reaction is occurring. You classify the reactions in many ways. Is it exothermic reaction? Is it endothermic reaction? Is it a reduction reaction or an oxidation reaction? That's when you start talking about what you refer to as redox reactions. So oxidation or reduction reactions. Different things that you talk about as far as reactions are classified. You talk about the reactants and the products in the reaction. Which components are reacting? And when they react, what is the product? You talk about the energy profile of the reaction. When the reaction is, before the reaction occurs, what amount of energy is required for the reactants to get to, say, for instance, transition state before they form a product. That's the energy profile that you are talking about. You talk about which conditions are necessary for the reaction to occur. Is the reaction going to occur in an acidic medium, or is going to occur in a basic medium, for instance? So, for instance, if you go into the next stage, you will see where reaction condition matters in biology, for instance. Now you move to the last stage where you see the merging between biology and chemistry. So in biology, you are now moving from that cellular level to the molecular level. So for instance, if you take the cell membrane, you now want to look at the molecules that make up the cell membrane. So you start looking at this cell membrane is made up of some phospholipids with some proteins embedded in it, or there can be some glycoproteins, glycolipids that are on these proteins or on the lipids serving as some receptors. So you are now referring to molecules. And what are the molecules you talk about? These molecules are either carbohydrates, or they are proteins, or they are lipids, or they are nucleic acids. So you realize that throughout, those of you coming from HND now, you realize that the biochemistry you were doing at all times when you were referring to the molecules or anything you were doing either practically in the lab or even theoretically biochemistry you were studying, it was about these molecules, what we refer to as the biomolecules, biological molecules, to be carbohydrates or its proteins or its lipids or nucleic acids, because these are the molecules that make up the living systems. Right. Now, if you are talking about molecules at this level of, biology, then of course, you can now talk about the interaction between the molecules. This is where you're now referring to biochemical reactions. So you have biochemistry. And these biochemical reactions come together to give you what you refer to as metabolism. What's metabolism? It's a collection of reactions that are required for the sustenance of life of a living system. Because metabolism is what? It's either breakdown of the molecules or the synthesis or the build-up of the molecules. So then in that aspect, just like you were looking at reactions, looking at reaction types, here too, these metabolic reactions, you begin to look at what type of reaction is it? Is it an oxidation reaction or is it a reduction reaction? Is the reaction breaking down a molecule or two or more molecules are coming together to form a bigger molecule? That's why I refer to catabolism or anabolism. Catabolism being breakdown, anabolism being build up, anabolism sometimes being referred to as synthesis or biosynthesis. You refer to the reactants, which molecules are reacting, and when they react, which product is it giving you? You refer to the reaction energy profile, which amount of energy is needed for the reaction to proceed. And at this stage, you start talking about things like enzymes that will reduce the activation energy so that you can have the reaction catalyzed. The reaction can be fast because the reactions have to occur in milliseconds and all of them coordinated because there are many reactions that occur. Imagine one reaction is occurring very slowly, the one which is responsible for my speech. Then it means that at a point my speech will become very slow and you have to take a very long time. Or maybe the reaction which is responsible for your hearing is slow. So when I speak, your ear will be hearing it slowly. The speech will be long. Or you want to just move your hand and the reaction which is responsible for contraction and relaxation of the muscles is that slow. See how long it's going to take for you to, do uh, the basic things of life. So this is where you have these enzymatic reactions coming in, and you have also the reaction conditions being considered at this stage. Your salivary amylase is acting at a particular pH in your mouth. When it moves into the stomach, the condition there is different. It will be inactive. But there, a different enzyme is active. So reaction conditions are also considered. So this is where you see biology and chemistry meeting to give you biochemistry at the molecular level. So, a lot of times biochemistry is shifted so much to, to be referred to as molecular biology. Even though, no, molecular biology, actually, yes, it is about these biological molecules, except that because one of the major discoveries in biochemistry has been with the structure of nucleic acids. And so there's that kind of bias towards studies of nucleic acids, DNA, studies, manipulations, uh, to be referred to as molecular biology. But actually, molecular biology is about these molecules, these biomolecules. So it's not only about the nucleic acids, but it's about all these biomolecules. All right, so I hope that this brief introduction should take out maybe any sort of thought of is it mystery or sort of making it look like this is kind of a difficult thing. So I hope that it clears your mind systematically, so you know what biochemistry is really about. Now we can move on to the business of the day, starting with fundamental enzymology. So the study of enzymes. So you see here, the enzyme is a protein. That's a biomolecule. Now, the first thing is, what are enzymes? What are they? Now, these are biological catalysts. They are responsible for the rate of chemical reactions. They increase the rate of chemical reactions. Now, most enzymes are proteins, as I said, but of course, you can also have some what we refer to as ribozymes. These are some catalytic RNAs. They are responsible for the metabolism of nucleic acids themselves. So like the breakdown of nucleic acids, you find these ribozymes that are participating in the process. because you have some form of recycling of biomolecules. You have these molecules called ribozymes, which are RNA molecules, but they can catalyze catabolic reactions of nucleic acids. They are not proteins, but most enzymes are proteins. Okay. Now, why do we need enzymes? Why do we need them? So there's a reaction that is on your screen. This reaction is a common reaction that you have known. You talk about it all the time that when you take in carbohydrates, the carbohydrates are broken down into carbon dioxide and water and gives you energy. So what you are seeing is this is sucrose, so table sugar, a common sugar that we use. And yes, in the presence of oxygen, it's breaking down into carbon dioxide, giving you water, and then it gives out some energy. It's a very simplified form of saying it because you know that there's different processes that this will undergo to generate that energy for you. But fine, that's okay. But the thing is, if you take this sugar, if you have a jar of sugar and you place it on your table in the kitchen or in the dining hall, do you see the sugar turning into carbon dioxide and water and giving out energy? No. Like you put the sugar down, you go out and you come back and the sugar is no longer there. The sugar is there. For a long time, you still see that the sugar is still there. It's not turning into carbon dioxide and water and energy giving out. And yet we say that when you take in this sugar is broken down into carbon dioxide and water and giving out energy. How does that happen? So you see, this reaction that you are seeing, it is actually occurring spontaneously, but it's occurring very, very slowly. It's negligible that you don't see that such a thing is occurring. Because if you have a jar of sugar, it will just be there. Maybe what you will see is that because sugar itself is hygroscopic, it can absorb water from the atmosphere. So maybe what you can see is that the sugar has turned into some kind of a cake. But if it is very little, just below the bottom, it may become a bit watery because it absorbs water if you open it down and things like that. But it doesn't turn into carbon dioxide and water, and you don't see it there again. Because the rate of the reaction is so slow that it is negligible. You won't see that. But you see, the moment you take in that sugar into your body, yes, this reaction occurs. And this reaction occurs in just some seconds, milliseconds. You have energy released to the cells. And that is because of the action of enzymes. We'll see the mechanism by which enzymes can do that to increase the rate of chemical reactions in that manner. Now, you see, if you were running some physical reaction in the lab, like so some chemical reaction in the lab, one of the things you do to increase the rate of chemical reactions is to apply heat. That is to give some energy to the reactant molecules so they react quickly and you get your product. It's a way of catalyzing reactions to increase the rate of reactions. You can just apply some heat. But this is not an option for biological systems. I mean, what's the physiological temperature of human beings, for instance? It's around 36.6 degrees Celsius, approximately 37 degrees Celsius. And you know what that means. If you increase the temperature of the body above that, say 39, 40, you know what it means. The biological system is made up of biomolecules like proteins, nucleic acids, and you know when you increase temperature up to a certain level, these biomolecules will begin to get denatured. So you can't increase the temperature of a biological system to increase the rate of the reaction. So you have enzymes to do that, to increase the rate of chemical reactions through other mechanisms, which we will look at. All right. Now, since we're talking about enzymes and the mechanisms by which they act, let's take a look at, basic principle by which all chemical reactions occur. Basic principle. I mean, this principle applies to all reactions, not only reactions that are occurring in biological system. And the principle is that you see where you are now, maybe sitting down, maybe lying down. At any point in time, if you feel uncomfortable, you will adjust yourself to a more comfortable position. Let's call that a stable position. Now, that happens also at the molecular level, that you have molecules that are at a stable, energy level. Now, molecules that are reacting, they originally exist at a stable energy level. If the energy level at which they were was not stable, then those molecules would not have been existing. So for instance, if you say something like water exists because it's having a stable energy level at which it exists as water. So all chemical substances have stable energy levels that they exist. Now, if two molecules are to react to give you a product, and originally they are already at a stable energy level, there will be no reason for which they will be forming something else, a product, because they are already comfortable where they are. They are stable where they are. So the analogy I was making from the beginning, if you are comfortable where you are sitting, and there will be no point for you to change that position. If you are comfortable, it is only when you are not comfortable that you will change. But if you are comfortable, there will be no point for you to change. So you continue to be in your comfortable position, in that stable position. So this is what it is with molecules, also with energy levels. So it means that for a reaction to occur, if you look at the energy profile on the left side, you see the reactants are at a certain energy level. On the vertical axis, you have energy. So energy level, you have energy level on the vertical axis. And just the horizontal axis, you have reaction coordinates. Now, where you see reactants at that energy level, they are stable. Now, before they will react to form a product, these molecules must be agitated to an energy level, which we refer to as the transition state. So it means that the molecule is agitated to a certain energy level that now it will begin to look for a more stable energy level. And because you have a reactant, so these are different molecules that are being activated, they can react at a transition state to form a product which is an energy level even lower than the energy level of the reactants themselves. Now, the blue line is representing the reaction profile of a non-catalyzed reaction. Here, because we are talking about enzymes, it's being referred to as a non-enzymatic reaction. And the orange line is referring to the energy profile of an enzymatic reaction, or call it a catalyzed reaction. And you see that the transition state of the catalyzed reaction is lower than the transition state of the uncatalyzed reaction. Why? Because you see that the reactants with the catalyzed reaction, so the enzymatic reaction, first of all, you see the enzyme is forming a complex with the reactants, which is an enzyme substrate complex. So that enzyme substrate complex requires a lesser energy to reach a transition state. And that is how the reaction can occur faster, because it requires a lesser amount of energy for a product to be formed. The same product which is formed with the uncatalyzed reaction, it is the same product which is formed with the catalyzed reaction. Now, what you need to keep in mind is that enzymes, or say a catalyst, only comes in to speed up a reaction which is already occurring. It is not the enzyme, or the reaction must already be occurring spontaneously, and the catalyst only comes in to increase the rate of the reaction. So yes, the same product will be formed, except that for the uncatalyzed reaction, it will take a longer time for the product to be formed, because the activation energy required to reach a transition state is much higher than for that of the catalyzed. So the catalyzed, the reaction will occur, faster, and you have your product faster. So this is a basic principle by which reactions occur. Now, before we go further, we need to understand some terminologies, because language, every field, every course has its terminologies. Finance people will have their terminologies, business, marketing. When you come to chemistry, biology, and specifically, we are doing biochemistry, and now this is enzymes. So specifically to enzymes, there will be some terminologies we'll be using, which you should understand, so that as we go forward, when we mention these things, you know what we are referring to. The first is cofactor. Cofactor. What's a cofactor? So we said from the beginning that enzymes were mostly proteins. But you have some non-protein part that is necessary for an enzyme to be active. And many of these are metal ions. Can be some magnesium ion, sometimes maybe iron 2 ions, etc. Zinc ions. These are metal ions that mostly you see they are attached to that protein, which is the enzyme. And their presence is required for the functional structure of the enzyme. It must be there to let the enzyme be functional. Then we have coenzymes. Coenzymes are also non-protein parts of the enzyme, but these are organic molecules. They are not metal ions. Well, in some biochemistry books, cofactor coenzymes are used interchangeably, but in some, they are actually distinguished. They have that distinction between them for you to know that a cofactor refers to these metal ions, inorganic metal ions, whilst the coenzymes refer to organic molecules like some derivatives of mostly the vitamin Bs. So when we're looking at coenzyme, you will come to see some of these vitamin B's derivatives of them that serve as coenzymes. They are required for the activity of enzymes. There was the apoenzyme. The apoenzyme is the protein part of the enzyme, the protein itself, sometimes being referred to as apoprotein. So you can have apoprotein, apoenzyme. It's only referring to the protein part of the enzyme. Then active site. So an active site, you see, a protein is a large molecule. But serving as an enzyme, there is a specific part of that protein where the reaction occurs. And usually this part is made up of like 3 amino acids that are in the peptide chain, but because the protein folds, these amino acids come close together. That it's like some 3 fingers that can hold molecules, the molecules that they are involved in catalyzing their reactions. So in those specific positions, you have the reaction occurring on those enzymes, the proteins. So it's not like the protein is large and at any part of it at all and a reaction can occur. No, there's some specific part of the enzyme that the reaction can occur. It is that particular part that we refer to as the active site. Then we have processes like activation. So activation, that is any process that is involved in increasing the activity of an enzyme. So you just maybe a metal ion which attaches onto the apoprotein will activate it. If the metal ion is not there, there will not be that activation to occur, like what we refer to as a cofactor. When a cofactor attaches, it activates. But sometimes even a substrate can be an activator. So like if the process of initiating or increasing the action of an enzyme is that activation, what does that is the activator. Then we have the process of inhibition, which is the opposite of activation. So any process that is going to render an enzyme inactive so that a reaction completely stops. It no longer occurs. Or maybe it's just decreases the activity of the enzyme. So it reduces the rate of the reaction. That is inhibition. And a compound that will do that is an inhibitor. So an inhibitor is the opposite of an activator. Then the substrate is going to be the compound, the substance on which the enzyme is acting to accelerate its reaction. That's the substrate. So if you have some reactants that are reacting, like we were referring to a reaction of, say, sugar. The sugar is a substrate, and an enzyme will be acting on it to accelerate its breakdown. So that's a substrate. So these are common terminologies that you hear when we are talking about enzymes. Now let's continue to look at some naming. How do we name enzymes? How do we name them? So enzymes are named in different ways. One of the ways that enzymes are named is just to add a suffix or the ending ase, A-S-E, to the name of the substrate on which the enzyme is acting. So, for instance, an enzyme which acts on urea, the breakdown of urea, is called urease. So if you see the name of an enzyme, urease, it quickly tells you that that enzyme is involved in catalyzing a reaction of the substrate urea. And this particular enzyme urease catalyzes the breakdown of urea. The same way you have lactase. Lactase. Lactase will also be acting on the substrate, the milk sugar lactose. So you see the name lactase is ending with ase. It's catalyzing the breakdown of lactose. So that's one way of naming enzyme. You adding the suffix ase to the name of the substrate. Another way of naming enzymes is based on the activity that the enzyme is catalyzing. So, for instance, you hear of the name DNA polymerase. Polymerase. And that enzyme, DNA polymerase, catalyzes the process of joining nucleotides, the basic units that form nucleic acids together, joining the nucleotides. So the nucleotides are monomers. It will join them to form the polymer, which is the DNA molecule or DNA fragment. So the process is polymerization. That's the activity that the enzyme is carrying out. So it is named after that activity, polymerization. So you see DNA polymerase. Another one, pyruvate decarboxylase. So think of decarboxylase as to say deleting of the carboxylic acid group, removing of the carboxylic acid group, the COOH group, removing it. You have seen these enzymes acting in different reactions before. So like pyruvate decarboxylase, you've seen it in carbohydrate metabolism. And you've seen DNA polymerase too a lot of times at the HND level. So you see pyruvate decarboxylase is removing carboxyl group, the COOH group from pyruvate. The substrate is pyruvate. Then another way is this time around, it is not adding that suffix ase, but it's rather adding zyme. from the word enzyme. So you see, the activity is the breakdown of bacterial cell walls, what we refer to as lysis of the bacterial cell walls. And the name of the enzyme is lysozyme. Lysozyme. So here too, you have the naming based on the activity of the enzyme, the activity that the enzyme is catalyzing. Now, apart from these two systemic ways that we have seen of naming enzymes, we see also some common names of enzymes. So, for instance, we see an enzyme named pepsin. That's a common name. It doesn't follow that systemic naming that we saw earlier. And this is just because this is one of the first enzymes that was extracted, and it was extracted from digestive juice. So it's been known to be involved in digestion. And Greek word for digestion is pepsis. So that name came, the pepsin came from that word pepsis, to tell that the enzyme is involved in the digestion of foods. Another common name is trypsin. And this name came from also the Greek word tryne. And this is because it's also one of those first enzymes that was extracted from pancreatic tissue. And the process of the extraction of it was by just rubbing glycerol on pancreatic tissue to extract the protein into glycerol. And because it was like you are wearing off the protein from the tissue into glycerol. That process, in the Greek word for it, is trying to wear down. And so the name was given to it, trypsin. Okay, so those are common names. Right. From nomenclature, let's move on to some classification of enzymes. Now we'll take a look at 6 major groups of enzymes. The first group we refer to as oxidoreductases. So this time around, this classification too is based on the types of reactions that the enzymes are catalyzing. And you see the first group, oxidoreductases. These are enzymes that catalyze oxidation and reduction reactions, what we commonly refer to as redox reactions. And an example that you see here is lactate dehydrogenase. In fact, if you read in a biochemistry book and you see the name of an enzyme with this dehydrogenase in it, you quickly know that enzyme is an oxidoreductase. Now look at what lactate dehydrogenase is doing. If you look at the reaction, so you see the reactant is, or the substrate is lactate, and the product is pyruvate. Now look, the second carbon in the middle is having a hydroxyl group on it, OH. There's an OH, that's a hydroxyl group on the second carbon in lactates. You come to pyruvate, and that hydroxyl group is gone. You now have double bond O. You have a C double bond O group rather there. The ketone group. That's what you are having over there. That's an oxidation reaction. Hydrogen is gone, and you now have just oxygen. It's an oxidation reaction. And the enzyme which is involved in catalyzing that reaction is lactate dehydrogenase. So lactate dehydrogenase is catalyzing the oxidation of lactate to pyruvate. The second group, the second class of enzymes we refer to as transferases. Transferases. And this is simple. It's coming from the word transfer. Transfer of what? Transfer of functional groups. So you have an example of the enzyme aspartate aminotransferase. Aspartate aminotransferase. So the name tells you aspartate. Aspartate becomes the substrate. Aminotransferase. So amino, so it will be transferring an amine group. That same enzyme is also called aspartate transaminase. So the aspartate is still there as the substrate. Transaminase is telling you the transfer of an amine group from aspartic acid or from aspartate. Aspartate is an amino acid. Now you see the reaction. The reactants you see here, aspartate, that's an amino acid or aspartic acid. You have alpha ketoglutarate. You see, when you go to the TCA cycle that you studied before, you see alpha ketoglutarate there as an intermediate. If you go back to, just go back and look at that TCA cycle, which you studied before, then you will see there is alpha ketoglutarate along the line somewhere. Similarly, you also see oxaloacetate also there. You actually see oxaloacetate in the very first reaction. Then you see glutamate is also an amino acid, which you studied. Now, this reaction actually, this is where you actually see that there is an interconnection between metabolism of carbohydrates and proteins. All the metabolic processes are connected. They are not just standalone on themselves. So when we continue, you come to see, when we go into the metabolism of these biomolecules, you see how all of them get interconnected. And this is just an example, taking a specific reaction, looking at an example of an enzyme involved in the transfer of functional groups. And which functional groups are being transferred here? Now, if you take aspartic acid and you look at the second carbon from the bottom, there's an amine group on it. It's an amine group there on aspartic acid. Now you take alpha ketoglutarate and you look at the second carbon from the top, you see there is a ketone group there, C double bond O, oxygen there on top. Now look at oxaloacetate. You realize that in place of the amine group in aspartic acid, you now have this C double bond O coming to replace. So just the double bond O coming to replace the amine group, and it forms oxaloacetate. And when you look at glutamate, it is the transfer of that amine group from aspartic acid to the place of where the double bond O was in alpha ketoglutarate. That gives you glutamate. So it's just an exchange. There's an exchange between these two, the amine group in aspartic acid and the C double bond O in alpha ketoglutarate. There is that exchange between them. And when that exchange occurs, you have oxaloacetate and glutamic acid or glutamate. So that's a transfer. The third class, hydrolase. hydrolase. So these are involved in the catalysis of hydrolytic reactions, hydrolysis. So we see an example, acetylcholine esterase. In the presence of water, acetylcholine esterase breaks down acetylcholine into acetic acid and choline. So the acetic acid portion on the left is broken off, gives you that acetic acid, and the choline portion is now separate. That is a hydrolysis reaction. It's a big molecule, acetylcholine, which is broken down into acetic acid and choline. This is a reaction which is involved in contraction and relaxation of muscles. So acetylcholine serves as some neurotransmitter giving signal. We'll come to, I think we'll look at some practical application of this reaction. Right. The fourth are lyases. And lyases catalyze reactions that either remove groups from molecule to form double bonds in the molecules, or it will add groups to remove the molecule, to remove double bonds from the molecule. Both ways. So these are, an example is aconitis. The reaction you are looking at is also is also one of the reactions that you see in the TCA cycle. If you go back and look at it, just pay attention and look at the TCA cycle, you see cis aconitate is somewhere and is converted to isocitrate. And that reaction actually is a reversible reaction. So you see, we say the lyases either will add groups to remove double bond from a molecule, or it will remove groups to add double bond. So if you look at cis aconitate, you see there is a C-C double bond here in cis aconitate. That bond is gone. It's now a single bond because there is an addition of water. The reaction here is a hydration reaction, where now you see from the water, OH is now bonded on this carbon. You see the OH there on this carbon, whilst the hydrogen from the water goes onto this carbon. This hydrogen and this OH were not in cis aconitate. That's why you have the double bond there. And the water molecule giving, that hydration removes that double bond from cis aconitate. The reverse of this reaction will mean that there's going to be removal of water to give you that double bond. And that will be dehydration. So that is what lyases do. Now, isomerases. This one too is easy from the word isomers. Isomers. And what are isomers? Compounds that have the same chemical formula, but different chemical structures. So you see, glucose 6-phosphate converted to fructose 6-phosphate. These two compounds are isomers. They have different structures, but their chemical formulas are the same. This reaction too, you see it in the glycolytic pathway. And the enzyme involved there is phosphohexose isomerase. The name even tells you that it is involved in isomerization reaction. And it is acting on a phosphohexose. What's a phosphohexose? A 6-carbon sugar with a phosphate group in it. That's a phosphohexose. Now we can go to the last class referred to as ligases or synthetases. This one too, you have seen DNA ligase as an example, particularly, after you have the formation of a new nucleic acid, from the process of replication, you have some gaps that are left, particularly on the lagging strand, and even in the leading strand, because you have the process being a semi-conservative process. So you have DNA ligase coming to fill in those gaps that are left between the nucleotides. So the ligases or the synthetases are just involved in catalyzing the joining of these nucleic acids. You say the joining of two molecules. So those are ligases. So how many classes of enzymes have we looked at based on the type of reactions that they are catalyzing? 6. These are oxidoreductases, transferases, hydrolysis, lysis, isomerases, and ligases. 6 of them. I think it's okay for now. We can stop here. The next time when we meet, we can continue. So for now, I've been talking for more than 1 hour, so almost getting to 2 hours. I will open the floor if you have any questions, any contributions. I know some of you have been working for years. You have some experiences, and so you can share them with us. So the floor is open. You can either write in the chat. When you write I'm monitoring the chat. Nobody has written there anything yet. You can write in the chat, or you can raise your hand, and then I will enable your mic. I call your name, you unmute yourself, and you speak. All right, so the floor is open.